Now it's my pleasure to introduce Dr. Jeremy Shefner, who is a neuromuscular neurologist at SUNY Upstate in Syracuse, New York, at least for now. And he's going to be talking about biomarkers and pharmacodynamic surrogate outcomes. And some are good.
Some are good, some are bad. And so Will assigned me this title, so I kept it. I'm under no illusions that an outcomes talk is going to be as dramatic as an early phase Clint Eastwood movie, but we'll go anyway. So Merritt began the discussion of outcome measures on Monday, and I'm going to focus on outcome measures that hopefully teach you something about the disease in question or the target that you're trying to address. And so just to review the bidding a bit, here's just a way of organizing thoughts about what kinds of outcome measures exist that can be used in a clinical trial. So the gold standard in general for approval of a drug remains an endpoint or outcome measure that's clinically relevant, which has a deceptively simple definition, basically how a patient feels, functions, or survives. And so that's what we aim to impact with any kind of drug or device therapy or medical treatment. And so that's what the FDA wants to see, if at all possible, before they're going to approve the drug. But many other endpoints are useful in the earlier phases of drug development, and some quite late as well. And so I'm going to focus on surrogate endpoints and pharmacodynamic endpoints, which are overlapping concepts. A surrogate endpoint is a value of some sort. It's a lab value, it's an image, it's a behavioral assessment even, that's intended to substitute for the relevant endpoint or to predict one in a very, very close and unbreakable fashion. So, and so it has a place in clinical trial development that we'll focus on a little bit. Related to that are endpoints that are known as pharmacodynamic endpoints, which reflect either a potential activity of a drug or therapy at a specific target, or affecting a specific pathway, or measuring the progress of the disease in a way that you can identify as clearly related to your intervention. The difference between surrogate endpoints, which clearly predict a relevant a clinical outcome, and pharmacodynamic endpoints are that in general, the relationship between clinical relevance and your pharmacodynamic marker can be much, much looser. And you don't necessarily need to have a predictable relationship there to make that endpoint useful. Biomarkers, which people talk about all the time, I realized as I was thinking about this talk, I don't actually know what a biomarker is. So Wikipedia says it refers to a measurable indicator of some biological state or condition. The NIH actually has a definition, which isn't much different. It's a characteristic that's objectively measured and evaluated as an indicator of a biological process, a pathological process, or a pharmacological process, or a therapeutic intervention. So what I've come to understand is that biomarker is anything that we think about related to a disease or health. And so it actually has no meaning to me, and so I'm not going to talk about it anymore.
Good decision.
So I'm going to focus on surrogates and pharmacodynamic endpoints because I think with that and clinically relevant endpoints, we span the entire spectrum of what we can measure. So that's the goals of what I'm going to talk about in the next few minutes are to illustrate uses and definitions of pharmacodynamic and surrogate markers, discuss ways they can lead you astray, talk about how and when they're useful, and a little bit, although I can't really focus on this too much, but what does it take to make a PD or a surrogate marker an effective part of your armamentarium? So one important thing to recognize is that surrogate markers and pharmacodynamic markers have different functions and play roles in different parts of drug development. So a surrogate is usually, although not always, it has its main function in later phases of development. because its intention is to substitute for a clinically meaningful endpoint, which is what you want to find in your late stage. You use it if it makes your life easier. So, people have mentioned, you've heard this sort of code word of underpowered stage phase 3 trials all the time. And so what the use of a surrogate marker is intended to have is to get you out of that dilemma of performing an underpowered stage 3 by giving you a marker that changes faster or more sensitively or allows you to use fewer number of patients, so you can get a signal in a phase 2-ish size trial that then can allow you to progress to stage 3, or in some cases to obviate the need for stage 3. If it's not faster, if it's not more sensitive, it doesn't do you much good. So because of the overlap between pharmacodynamic and surrogate markers, many surrogate markers also give you some insight into mechanism of action, but they don't have to. And the risk or the limitation to use of surrogate markers are pretty multiple, and I'll talk about some of them. But a big problem is the strength of the relationship between the surrogate and the clinically relevant endpoint, and the validation of your measure, which I'll discuss next. So, in contrast, pharmacodynamic endpoints are usually thought of as having a main use earlier in development, and often, though not always, they have something to do with what you understand about the mechanisms of action of your intervention. So you can measure target engagement. You can measure activity along a certain pathway that you think, although you're not certain, is related to your disease, it's a measure that can be a fluid marker, or a physiological marker, or an imaging marker that changes with disease progression, but in a more rapid fashion than your clinically relevant endpoints. And so here's just a bunch of endpoints that I've put into this Venn diagram. You could argue about moving them in various ways. On the right, are things that are clearly pharmacodynamic. They can be imaging, such as a PET ligand for A2 receptors in Parkinson's disease. They can be inflammatory measures that assess a target that's related to the inflammatory system. They can be measurements of levels of proteins, such as NOGO-A in ALS. And then in the intersection between the two are things that are measures like that, but for which we really have a lot of confidence that they have a direct impact on disease, and measuring them is almost as good as measuring an outcome that's directly related to health. And so, for example, probably the most striking one is HIV viral load, which we now know is not only a good thing for it that does demonstrate drug activity, but has an impact on health of an individual. And so it's both a pharmacodynamic marker and a surrogate. Farther to the left are things that maybe are clear predictors, but don't necessarily give you much insight into the pathway that impacts those measures. And then on the bottom are things that you could stick in there, but really are more or less recognized as being clinically relevant. So both surrogates and pharmacodynamic endpoints sort of feed down into measures at the bottom, which are the gold standard for demonstrating that your intervention has some benefit. So just another list of things that have been used in the past as surrogates that are, and to some extent have, at least in the first few, have been able to be used to get drug approval in the United States. So glycosylated hemoglobin is a measure that many diabetes trials use as an approvable endpoint for clinical trials. Cancer studies can get a drug approved based on shrinkage in tumors. CD4 count has been used in the past for AIDS studies. Then when we get down to neurologic studies, we're less clear about where these measures play a role in the drug approval process, but MS plaques are measured very well. We have all sorts of radioligand imaging techniques now for Parkinson's disease, and then measures such as measuring the number of motor units. I know that it was on the slide that Eric used, motor unit number estimation, because I gave her that slide, but I don't think she talked about it. But it's a physiologic measures of motor unit health. Transcranial magnetic stimulation evaluates motor pathways. So those are things that potentially could be used in the appropriate circumstance to justify either earlier approval of a drug or going on to a phase 3 study with clinically relevant endpoint. So this is a slide that I believe James Berry gave me that sort of shows where pharmacodynamic markers can be interposed in clinical development. And so on the top rectangle, this is the relationship that both some pharmacodynamic markers and any good surrogate should have to a clinically relevant endpoint. And that is, you have a disease state, you have a marker that then directly predicts the clinical endpoint. And so that could be a pharmacodynamic marker, that could be a surrogate. That's where a surrogate plays a role. The other, as you go down, there are several other possibilities. So that you could have a disease that impacts clinical status in several different ways, several different downstream paths. And your pharmacodynamic marker could be very useful just at looking at one of them. So could we have the lightsaber?
Okay. Lightsaber?
Yeah.
You can throw it across the room and hand
Yeah, yeah. Thank you. Beautiful.
When you turn on
So in this example, one pathway is measured by the pharmacodynamic marker. The disease may progress along another pathway, or you might impact that disease along with a completely different pathway. But nonetheless, if your drug is intended at impacting this pathway, that's a very useful thing to be able to measure. Down below, you actually may have a marker that's sort of an epiphenomenon that has absolutely no direct relevance on how a patient does, but is impacted by the disease process often enough that it might be useful to measure and tell you something about how the disease might be impacted by your therapy. And then you can have this inverted so that what you're measuring is an impact of clinical status. And so, for example, an example of this might be serum chloride in ALS. So in ALS, people have more and more trouble breathing as they go into respiratory failure. Serum chloride changes. You could be measuring serum chloride. It's really reflecting not the disease, but the organism's response to the disease. Doesn't mean it's useless. It could be very effective and very sensitive and help you in early phase development. So surrogates really are intended to follow that first pathway, so that you intervene, you have a disease, the surrogate changes, and along that pathway, that impacts the actual disease, the clinical endpoint. But surrogates are complicated, and so this is, even in that pathway, there are all these other ways that things can happen in ways that you don't anticipate. So that your intervention could do something completely independent from the surrogate, which would have either a good or a bad effect on the organism, on the patient, but your surrogate might not reflect that in any significant way. You could have completely irrelevant things that are much more important than your treatment and not measured by your surrogate, which also impacts the clinical status of the patient. And so many things can happen to change the relevance of your surrogate as related to its impact on your clinically meaningful endpoint. And so there are some really sort of terrible examples of how surrogates have led people astray in drug development. This is a famous one. It's a cardiac study. It's been known for a long time that after MI, lots of arrhythmias are a bad sign. And so people who have lots of arrhythmias in the days after MI survive much less well. And so these drugs were approved by the FDA on the basis of measuring post-MI ectopy, with the assumption that reducing post-MI ectopy would reduce the mortality after MI. And they were approved, and they went to post-approval marketing surveillance studies. And it was determined that, in fact, although they did effectively suppress post-MI ectopy, survival was negatively impacted through a completely different pathway. And so they were ultimately pulled from the market for that use. So an example where the relationship between the surrogate marker and the clinically meaningful endpoint can be separated that you don't necessarily predict. So how do we use surrogates? And so the FDA has given us some guidance in this in terms of when and where surrogates are appropriate. And so in 2012, the FDA established 3 new pathways that drugs could be approved along. First is fast track, and then breakthrough and accelerated approval pathways. Fast track that was included because that was part of this 2012 advancement of the FDA. It doesn't actually deal with questions of surrogate or pharmacodynamic markers, but just to briefly talk about it, fast track approval can be obtained for a therapy that treats a condition that isn't otherwise treated, or has a significantly improved effect on a serious disease that isn't well treated, or can be used to treat a disease where there are significant numbers of patients who don't tolerate existing therapies. So you can get the general idea of when a drug can go for fast track approval. But that doesn't say much about how the drug is approved. It's usually approved through a traditional manner, meaning that a clinically relevant outcome is required. Breakthrough therapy specifically talks about surrogates. And so you need to have a serious condition, and you need to have a drug that potentially has a substantial improvement over existing therapy. And to that end, if you meet those criteria, an effect on what's called an established surrogate endpoint can support drug approval. And what they say that's useful if this surrogate endpoint is considered reasonably likely to predict a clinical benefit or a clinically relevant outcome. And so that's where the sort of ambiguity of when you can use it is. When does a surrogate endpoint actually can it be considered reasonably likely to predict an outcome? Accelerated approval is very similar. Again, it's limited to diseases that are serious conditions and with a clear unmet need. And so a surrogate endpoint can be used under these circumstances as well. But acceptance of the surrogate depends on the scientific support for that endpoint, which again is the sort of the ambiguity of when you can use this. And even under these circumstances, while that use of that surrogate can support approval, you then have to do subsequent studies after FDA approval with a clinically reasonable, clinically relevant endpoint. And so, what is an established surrogate? What is a surrogate that's reasonably likely to predict a clinical benefit? What do you have to do to make that happen? Make the FDA think that way? And so that brings up the concept of validation of markers, and this applies to both surrogates and to pharmacodynamic markers. And so, one thing, and this is the first aspect, is something we don't think about that often in academic centers. But if you wanted to establish a marker to be marketed as a surrogate, you have to. And so you have to really talk about, if it's a fluid, exactly how the fluid is handled. You have to establish that it can be done in a reproducible way in multiple centers. All are certified as being able to perform clinically based laboratory assessments. That both the handling of the material it can be standardized in a way that the reproducibility of the measure is very good. Then you go to aspects of your marker, which have to do with how does it change with drug activity, and what else can change it. So can change in diurnal cycle change your marker, can hydration, can diet, all those things have to be looked at. And then how specific that marker is also an aspect to getting your marker validated. So we've talked about this. And so I thought I'd just go through some different types of markers and talk about where they play a role in drug development. So let's talk about MRI imaging and multiple sclerosis. So here is a phase 2 study of BG12 or fumarate in multiple sclerosis. Relatively small study for MS, 257 patients divided into 3 groups, 6 months of treatment. And the primary endpoint of this study was gadolinium-enhancing lesions. And so this was, This is not a clinically relevant endpoint. It's a phase 2 study not intended to lead to immediate drug approval. But it's clearly, the goal is to impact a clinically relevant endpoint. And relapse rate is one such endpoint. And so here's a summary of the data. They had a dramatic effect on their surrogate endpoint, or pharmacodynamic endpoint. So a 69% reduction of lesion density at 6 months. They had a trend towards a lower relapse rate, but no dose response, and an average risk rate that was significantly was less than what they saw on the gadolinium-enhancing lesions, and not statistically significant. This is the data from the relapse rate, from the gadolinium-enhancing lesions, and you can see a pretty dramatic dose response that ends up in a massive reduction in lesion density. So the goal, this is a useful measure to the extent that it predicts an outcome. And so potentially it's very useful because it's much more sensitive than the clinical measure that they use. But that implies that if you did a bigger study, that clinical measure should show a significant effect. And in fact, it did. Here's the phase 3 study. Again, there was no dose response. This is the 2 doses of PG12. So the earlier study predicted the later study very well, but there was a dramatic reduction in relapse rate as compared to placebo, which is bad. So in this situation, that marker predicted a clinically relevant endpoint in a very effective fashion. And similarly, in another monoclonal antibody study for MS, this is actually a very small study looking at a single treatment of eculizumab, I think in about 20 patients, and showing that the new lesions after administration dropped very dramatically in a small study, not enough to really see any clinical benefit whatsoever. So they went to a larger study where they looked at this antibody in conjunction with interferon and showed a dose-dependent decrease again in enhancing lesions, but a trend that was not significant in relapse. So very much analogous to the PG12 study. With data, here's the 3 dose response curves for placebo plus interferon, placebo plus low dose, and placebo plus high dose of eculizumab. So again, a marker that shows a dose response that suggests, although doesn't prove a clinically meaningful endpoint. And just as a review of this, so there are a variety of ways to look at MR, and so enhancing T1 lesions apparently correlate with relapses, enhancing T2 lesions tend not to. So there's to give you some idea that there is a lot of complication in what you might think is something that should be directly related. So how would you classify gadolinium-enhancing lesions as a marker in MS development? Is it a pharmacodynamic marker? Well, it clearly seems to be related to disease progression. It clearly seems to be, at least in these circumstances, more sensitive to the clinical outcome. It's not pathway specific, so it doesn't fulfill all the goals of a pharmacodynamic endpoint. It's very much related to the disease rather than to a specific pathway that you're impacting. Is it a surrogate? Well, potentially. It's probably not a surrogate that the FDA would accept happily, but in these cases at least, very much predicted something that was clinically relevant. So it's a lot less clear when you look at Parkinson's disease and radio tracers, and I'm not a Parkinson's guy, but here's a bunch of different ways that you can use PET to assay different aspects of the basal ganglia and extrapyramidal function. You can look at fluorodopa PET that measures dopamine synthesis. You can look at SPECT with CIT, which measures dopamine activity at the level of the synapse. A whole host of things that you would think, given our knowledge of the etiology of Parkinson's disease, are both important for determining where your drug acts in the system, but also potentially clinically relevant. But here's a pretty famous study. This is the 2004 study that looked at impact of levodopa, the oldest Parkinson's drug, and disease. And so this was actually a a 40-week study where patients were actually treated with placebo, which is surprising to me, but also various doses of Cinnamon. And the UPDRS, when the numbers get higher, patients get worse. So you can see that this is the placebo function. Patients did clearly better at 150 milligrams a day of levodopa, better yet on 300, better yet on 600. Then they were all withdrawn from medication. And the UPDRS, which is the Parkinson's rating scale, all got a little bit worse after withdrawal. But long after the half-life of levodopa is over, patients on placebo were still doing much worse 2 weeks out later than patients on any of the treatment groups. So which suggests that levodopa actually had a long-range beneficial effect on Parkinson's. But they also looked at dopamine nerve terminals using beta CIT and found that there was a clear and dose-related trend towards worsening as measured by SPECT. So here's a marker that is relevant. It has a clear relationship to targets that we would think are important, but doesn't relate to clinical outcome. So, and if you look at multiple Parkinson's trials, you get very similar kinds of discontinuities between clinical outcomes and tracers. So the pramipexole study that actually led towards approval of pramipexole had a clear effect on the clinically relevant endpoint, but the reverse effect on the biomarker. The Ropinirole study was similar. And so here's a field where we think we know the pathway of Parkinson's disease. We have all sorts of markers that look at the effect of agents against that pathway, but don't necessarily predict outcomes. And then a final one that I don't have a punchline to. So adenosine receptors are now a target for potential therapy. Here's a PET scan which looks at adenosine receptors, and you can see that a drug that is supposed to block adenosine receptors, when given, reduces the uptake of this ligand that binds to adenosine receptors. So you can see that here is a quick marker that shows target engagement, but we don't know whether that has a clear effect on a clinically relevant outcome. And let's just say that again. So where does the radioimaging stand in terms of drug development in Parkinson's disease? Would you call this are they pharmacodynamic markers? Show of hands. Yes? No?
Left hand's for no, right hand's for yes.
So sure, yes, the answer is yes. So they effectively and reproducibly show you aspects related to target engagement on pathways. So that's all you expect if you have a drug that is intended to impact the A2 receptor, and you want to show that drug gets there. You can use that radioimaging technique to prove that to yourself. And so in early phase development, that could be very useful. So it's definitely a pathway-dependent pharmacodynamic marker. But just as clearly, these are not surrogates for outcomes. At least as far as we know, for the duration of the studies that have been done, a particular effect on any of these markers does not predict a specific clinical outcome. So I want to pull back a little bit and just talk about just some outcomes in general, and whether they are pharmacodynamic or surrogates or clinically relevant. And so here's a set of pathways that have to do with direct muscle activation. So here's a cartoon of the sarcomere. There are a couple of drugs that have been developed to impact the sarcomere. This was one that's been recently studied in ALS. It increases the efficacy of individual muscle contractions. This one binds to myosin pig filaments and actually is specific to cardiac muscle. And so looking at the cardiac drug first, see if I can make this go. Here's somebody in heart failure. On the left is untreated, on the right is treated.
There's like a little, you go over it and hit that little play button.
So, can you tell, can you look at, if you look just, not horizontal. All right, so we'll do it one more time. If you look at the left and look at the right, you can see, I hope that on the right, the valve is moving much more extensively, and the chambers are actually smaller. So show it one more time. And so there's a number of things here. There's just an echocardiogram, which is a marker for target engagement, because this is a muscle protein. It's also potentially a marker for disease status. If you look at stroke volume at the bottom, you can see that stroke volume with drug changed from 23 milliliters to 54, and ejection fraction increased from 18 to 23%. So lots and lots of different things you can measure that impact potentially other organs. So what are these things? So what's the measure? Are these clinically relevant endpoints? Show of hands. Yes, no, yes. No, they're not. So what a heart failure person cares about is not, what's my stroke volume? They care, how do I feel? Will I survive till next week? So at their best, they're still not clinically relevant outcomes. Are they surrogates? Well, we would have to know more about cardiology than I actually personally do. I mean, is there a way to keep somebody alive longer without increasing their ejection fraction? Well, my guess is yes. Conversely, can you increase somebody's ejection fraction and still make them more vulnerable to death? My expectation is probably yes, too. So they're probably not direct surrogates. But they are pharmacodynamic markers, because they clearly show that this drug had an effect on muscle that potentially shows target engagement and potentially shows a change that's relative to the patient. So there's a similar drug that has the equivalent effect on skeletal muscle. And so just this is the animal data. So you can take a nerve muscle preparation, stimulate the nerve, and the muscle force increases with the rate with the frequency of your nerve stimulation. Those are the light green bars. The dark green bars are what happens when you have this muscle activating drug on board, so that at sub-maximal contraction frequencies, there's significantly more force. And you can imagine a whole host of diseases where that might be a useful thing to have happen, including ALS. This is sort of showing the exact same thing in human volunteers, that if you stimulate the peroneal nerve, measure ankle dorsiflexion, with this drug on board, you get an increase in overall force at these contraction frequencies. The drug was the subject of a relatively large phase 2 trial with 400 subjects studied for 3 months, and one measure was vital capacity, the extent to which you can exhale after a maximal inhalation. And this drug showed a slowing of decline in vital capacity of about 70%, significant at a very high level, 0.0006. So vital capacity is a measurement of breathing. Breathing gets worse in ALS. Breathing is important for survival. Is it a surrogate? Is it a pharmacodynamic marker? What is it? Is it clinically relevant? One other piece is that strength may be important, but it doesn't necessarily predict outcomes. So here's a study from 2004 looking at patients with fast decline in vital capacity. They survive a lot worse than people with slow decline. But people with fast decline in extremity muscle strength actually survive better, not statistically, in this study. So that muscle decline isn't necessarily predictive of ultimate survival in ALS. So given those data, what is vital capacity, what is it as a measure? Is it clinically relevant? Is it a clinically relevant measure? Anybody? Yes. Oh, everybody who says yes. Everybody who says no. This is an interesting question, because this particular company is in the process of talking to the FDA about whether they can get their drug on the basis get their drug approved on the basis of slowing progression of vital capacity, with the clear expectation that a better vital capacity is good for you. And so I guess the answer is I don't know yet because the FDA hasn't said. Is it a pharmacodynamic marker? Well, I think clearly it is because it shows target engagement. And is it a surrogate? Well, that's the question also that the FDA has to decide. All right, so I think I have 5 more minutes to talk about. So these are, I've talked about imaging, I've talked about radio tracer imaging, I've talked about functional measures. What would be easier in almost any drug is if you could draw some blood or take some urine or do something with a fluid sample and get something important out of it. And so, when people talk about biomarkers, often that's really what they're talking about. Some fluid that will give you a signal that will allow you to do a really short trial or study far fewer numbers of patients. And so this is a potential fluid marker in ALS. Neurofilament heavy chain, which can be measured in the CSF, it can actually be measured in blood as well. And so this is from a paper written by Bob Bowser's group who studied fluid markers in ALS, showing that in the CSF, there tends to be a lot more neurofilament heavy chain in ALS patients as compared to ALS mimics, as compared to other neurodegenerative disorders, as compared to controls. And that more or less, if you measure people over various aspects of their disease, there is a correlation that suggests that levels change over time in a somewhat predictable fashion. So, what are the questions you would have to ask or what's the pathway you have to follow to determine whether this is going to be useful in drug development? Well, one thing is, while this is nice for a paper, it's no good having a marker where some normal subjects have the exact same measure as some patients, right? So as a diagnostic marker, in a population setting, it's sort of useful. In an individual patient, it has no benefit whatsoever. And similarly, although it's nice to have a little bit of a correlation with disease progression, this is clearly less useful than functional rating scales or vital capacity measurements or strength measurements. The variability here is much greater. So this just goes to, well, what do you need for a fluid to make it useful? Well, you need to be able to clearly distinguish one group from another. And you need if you want to use it as an outcome measure in a clinical trial, you need to have a rate of decline which is far greater than the variability, which is not the case here. So that's not to say that this can't be refined in some ways, but this gives you something that is not useful right now in terms of use in a clinical trial.
Well, is that study cross-sectional, though, or is it longitudinal? It might be that within subject variability is much less.
You're right, and so that actually was cross-sectional, and that's the part of the next study that's being done. So that's yes. So the answer is we don't know the answer yet in terms of the utility of this marker, and it is probably true that there are huge individual differences, and there are also huge diurnal differences. It turns out in measurement. So when you draw the CSF. What level of activity the day before actually it makes a difference. So there's all sorts of ways that you can make it more reproducible potentially. And so those are the next steps. It's how can you abolish the overlap if you want to use it as a diagnostic marker? How do you actually reduce the variability of the change over time? And then if you're actually interested in forming a company around this measurement, how do you actually develop labs that measure this in a reproducible way in clinically regulated situations. So the way where that came from was just a huge screen of lots of things that were looked at in ALS. So that was NFH was identified in a non-hypothesis-based method. You just looked at a bunch of stuff and saw what was different. Fluid biomarker development or fluid marker development can also be hypothesis-based. And so, again, just focusing on ALS for a bit, there is increasing evidence that a variety of neuroinflammatory processes are relevant in ALS. There's evidence for microglial activation. There are increased monocytes in brain tissue that were in areas of brain that's damaged by ALS. And you can measure increased cytokines, including IL-6 and TNF-alpha. So there are lots of things that you would measure based on your idea that inflammation is important in ALS. So not that you do a random screen, but you look specifically at inflammatory markers. And so this is a study from an MS group who started looking at ALS because of this interest in inflammation, showing that monocytes exhibit a specific inflammatory signature in both animal and human models of ALS, and that this signature potentially could serve as a marker of drug activity. This is irrelevant to that. This is looking at the mouse model of ALS and showing that in the spinal cord, but not the brain, there are increased numbers of inflammatory monocytes. This is pre-symptomatic 9 days, 120 days, 130 days, which is when the animals actually die. So there's this increase in numbers of monocytes with disease progression. And James Berry, who works with Merrick at the MGH, has looked at this in ALS patients and found that, again, this is just cross-sectional, looking at healthy controls, sporadic ALS, and familial ALS, there seems to be a reduction in CD14 and 16 positive inflammatory cells in the serum of ALS. So what is this as a marker? This is a potential diagnostic marker. It's certainly not useful in its present form, given this incredible overlap between normal and diseased patients. And just sort of similarly along this theme, T regulatory cells also appear to be different in ALS patients. Rapid progressors are somewhat different than slow progressors. Again, these are the kind of markers that you could look at based on your hypothesis that there's an inflammatory component to ALS. And finally, you want to get more specific than that. And so here's an example of a drug development program with a potential very specific biomarker. In about 2% of patients with ALS, they have a mutation in the SOD1 gene. There is now an ongoing program looking at antisense reduction in SOD1 in patients with familial ALS. And so if you could measure SOD1 levels, that would be a specific marker for target activation, potentially a surrogate for efficacy, although we don't know. So these are animal studies showing that antisense injected into the CSF reduces the amount of RNA related to SOD1 in the central nervous system. Correlated with that, there is a survival benefit in mice. A phase 1 and now phase 2 program with this antisense has been started in humans, where a biomarker, a pharmacodynamic marker of target engagement will be recording SOD1 levels in the CSF. So at the very least, if that shows a signal, that will be a signal of target engagement. Will that predict better outcomes? It's completely speculative, so it's not an effective surrogate. So, summary of what I've been trying to stress is that surrogate and pharmacodynamic endpoints can be overlapping concepts, but they have different functions. They're useful to the extent that they are better predictors than clinically relevant endpoints and give you an answer in a shorter period of time with smaller numbers of subjects. The pitfalls are many, and they relate to the relationship between clinical meaning and whatever you're measuring, variability of the measure, and how amenable whatever the pathway it is for potential treatment. But to avoid the conundrum or the obvious bad outcome of underpowered phase 3 studies, we need to develop these kinds of markers to allow us to move from early phase development to a late phase 3 evaluation.
This question may be better addressed to Scott Jones, but
No.
you know I can't answer.
Okay, let's take a break.
So this course is focused on clinical trial design, but I think a lot of us in this room are also interested in biomarkers because we realize that we'd be able to conduct conduct these studies a lot more easily if we had better biomarkers. And so I guess I'd just be curious to hear how people go about developing biomarkers and how the NINDS feels about this process. I know that there's a biomarker aspect to StrokeNet now, for example. So is the idea people would go through the normal mechanisms to get funding for that, and then StrokeNet would be a way to disseminate, to try to validate biomarkers? It's just not clear to me how that all pieces together, what the priority is at NINDS.
Well, let me answer that general and then redo the NIH piece. So marker development. So I think one of the things that I thought was lost a little bit in this discussion of networks is that even in the network, projects are PI dependent. So the networks don't determine we're going to do this biomarker study in LS. They are potentially receptive to somebody saying, I want to do a multi-center biomarker study in stroke or whatever. Early phase, it depends, do you need a multi-center kind of network to do pharmacodynamic marker development? Well, maybe. It depends on how common your disease is. So you need lots and lots of samples. And so in ALS, we have a group, the Northeast ALS Clinical Trials Consortium, which has 100 sites. And we've done studies that are tissue collection studies based on a principal investigator's project. Those things are the second study after somebody's own internal collection of specimens from that disease show a trend. So I think biomarker, shouldn't be talking about it, so marker development follows a similar progression as clinical development in that you start with a very small number of samples, which at least in some cases, and then you move to a more standardized multi-collection, multi-center collection, and then you potentially move even farther. A clear counterexample to that are genetic studies, where there's no point in doing small samples. You have to study huge sample sizes, and so you can't even think about it until you have your consortium in place. What the NIH thinks about it?
No, I always bounce things back because it's not what the NIH thinks about it. It's an academic problem. And so the big challenge is that a lot of these things that come in, Jeremy's absolutely right. In the sense is that if they are presented as fishing expeditions and the science, the justification behind the science is weakly formulated, it's not going to get through the peer review who's really looking to see whether or not the problem has enough understanding and the methods that are proposed are going to get to some answer that yields the development of the next step. And so I think it's not that it's frowned upon because it fits within the scope. We've tried different approaches to support these types of programs. Linking them to clinical trials. We've supported many ancillary studies that have allowed studies to be done within clinical trials, and I know how frowns upon them, but there are other opportunities in which I think they become great value. But it does come down to the rigor. I think that aspect, and if I can, Hal's not here to defend himself, so I can speak up. But why he frowns upon them is because we would often see applications where somebody would write a trial and they'd say, "Oh, I'm going to collect this," and it'd be 2 sentences about this biomarker. And this gets to that problem, is that it's not laid out. But that's an R01 application. StrokeNet built it in is an opportunity for that latter phase that is part of, are we at the point where we understand enough to really get it to where it's usable, and it needs a lot of sense. That is what the StrokeNet is built in. But otherwise, it is very much an R01 application. It can be justified, then yeah, there are many programs.
And I will say that, one of the more successful things that I've seen is if you have a larger study, and you can do it at some sites. So if there's a larger study and you want to collect biomarkers, there may be some sites where that's ideal to do it in, some sites that can't do it. I know that Shine did that, and that's the one that I know most recently. But I mean, it's an easy way to do it, and you can even pitch it to, depending on what the numbers are, just at your site alone. Is that correct?
Yeah.
I'd also add that fluid and tissue collection maybe seems easier than MRI, but it actually isn't. So what we found in our ALS consortium is that if you want to collect something as simple as CSF, you need to collect it in a very rigorous way. Every site has to do it exactly the same. And it has to be out of refrigeration for the exact same period of time. It has to be spun exactly at the same time. Same thing for blood. And so in many ways, if you can, it's much simpler to start out doing it at your own center where you can have control over these things, develop the protocol for handling the specimen, and then go to a larger format.
And I'll just throw one other thing out there. I mean, you could say that we can collect some things at a single site and try to, sort of see what we can learn there. You do stroke. So you need, you could say you can have a sample size, but whatever sample size you have, you need outcomes. You need the ability to find some degree of separation between, sort of assay sensitivity. And anytime I think you look at a biomarker and what it might predict, or whatever, I think, are you using all the existing information, and what is this going to add to the existing information? I mean, you call NIH stroke scale over time as a potential biomarker of response to TPA. How often is that even done? And I think if your biomarker is not more predictive than the NIH stroke scale in the outcome, then it's not adding any useful information. So I think thinking about how the biomarker is going to add to what you already would know about the patient is important, because it's got to add information to be useful. Because there's a lot of things that are like correlated with severity, and say, oh look, this is correlated with severity, but does it predict things better than the initial baseline severity or the change over time? So you have to. It's hard to disentangle, so you just have to realize you need events in both groups, people with the biomarker without.
But early on, you don't need to ask that question.
Right.
The first question is, does this marker behave in a way that is related to your disease? Then your next step is asking, well, does it get me anything that is better than what I've already got? Which is what Will is pointing out. But I mean, that's sort of a next phase question. And so again, you need outcomes for that later phase more than for the earlier phase.
There's a different set of statistical tools that are required. So, I mean, most statisticians we have training in them, but we don't necessarily do it on a regular basis. So, you want to be upfront about that you're not just trying to show that an association exists, because that's a lot easier than showing that this actually predicts the outcome that you're looking for.
And I guess the other thing to add is, like in this pipeline as things are developed, right? I mean, the problem that we have is we only have so much money that can be capped. So there's lots of great ideas, but unfortunately only a few can get chosen because it goes up to our line. But there are other resource pathways to help support these earlier developmental and preliminary data phases that bring things to the next level. I mean, you have to go out and you have to build from what's been done to start to put it together, but there's, in order to sort of rise into that territory that's competitive within NIH, there's got to be enough behind it to sell it to a group. If you can't convince your peers, then it's going to have to come.
And marker development actually is sort of a common area where entrepreneurship is employed.
So
if you think about those radio tracers, virtually all of them have sort of morphed into small biotech companies. That NIH study is now the nidus of a small biotech company looking at that. So that another aspect.
So there's an FDA guidance on the development of companion diagnostics. So that might be something that you should Google and think about, because I think if you're looking at this idea of are you saturating the target, that may be an area of interest for a pharmaceutical company that is helping to maybe developing a more early phase molecule with. So I think you might want to read that document. It's kind of interesting.